Today, we're going to dive into a really fascinating story from the front lines of cancer research. It's a story about how scientists figured out how to turn a leukemia cell's greatest defense into its ultimate downfall.
And this whole story, it kicks off with a single number, 30%. That's how many cases of a common blood cancer, acute myeloid leukemia, or AML, are driven by one particularly nasty genetic mutation. And yeah, it's a mutation that has made this cancer incredibly tough to treat.
So, this huge challenge got researchers thinking, and it led them to ask a really clever question. One that basically reframes the entire problem and sets the stage for a whole new way to fight back.
All right, so let's meet the bad guy in our story, a specific and very stubborn form of AML. So the whole problem here boils down to this one faulty gene called FLT3. Now, in your healthy cells, this gene helps keep growth under control. But when it mutates, it's like a gas pedal that's stuck to the floor. It's just constantly screaming at the cancer cells to grow, divide, and survive, no matter what. And that's exactly why patients with this kind of AML so often relapse, even after really intensive treatment.
Now, if you want to stop something like this, you first have to figure out how it defends itself. And that search led scientists to a pretty interesting discovery about the cancer cell's metabolism. As researchers were studying these FLT3-mutant cells, they noticed something weird. The cells were in total metabolic overdrive, but specifically when it came to making lipids, fats. It was like each cell had its own tiny factory just churning out fat molecules 24/7. Which, makes you ask the obvious question. Why? Why all the fat? What's it for? Well, it turns out this lipid production wasn't just for energy. The cell was literally creating a kind of protective suit of armor for itself. Okay, so the next piece of the puzzle was figuring out who was running this factory. How was that faulty FLT3 gene actually giving the orders to build this armor? And it turns out the key middle manager here is a protein called EBPα. It's a type of protein we call transcription factor, but honestly, the best way to think of it is as a factory foreman. Its whole job is to get signals from the higher-ups, and in this case, that's the faulty FLT3 gene, and then go and turn on the specific machinery needed for a job. So you've got to picture this chain of command, right? At the top, you have the faulty FLT3 gene. It's the broken boss, just screaming, go all day long. The CEBP-alpha foreman hears that order, runs down to the factory floor, and starts flipping all the switches on the machinery, these enzymes like FSN and SCD. And just like that, the cell starts building its protective lipid armor.
Okay, so once you've mapped out that entire production line, you can start thinking, how do we shut this thing down? And that's exactly what some current treatments already try to do.
So there are these drugs out there, like gilteritinib, and we call them FLT3 inhibitors. Their whole job is to go right to the top of that chain of command and cut the power from the broken boss, that mutated FLT3 gene. But here's the cool part. Studying how these drugs work is what revealed the key to an even better attack.
And this right here, this is the big aha moment. When a scientist treated the cells with one of these FLT3 inhibitors, they saw exactly what they expected. The FLT3 signal turned off. But then they saw that the C/EBP-alpha foreman also went inactive. And because of that, the entire fat armor factory just ground to a halt. They had just confirmed the whole chain of command, from top to bottom.
And this wasn't just happening in a petri dish. To see how this all works in a real living system, the researchers used something called patient-derived xenografts. They literally took leukemia cells from human patients and grew them in mice. This let them test the drugs and prove that, yes, shutting down that boss gene really does dismantle the cell's lipid armor.
Okay, so this is where the story takes a really, really clever turn. Because taking away the cancer's armor doesn't just leave it exposed, it exposes a very specific and very fatal weakness. This weakness is a process called ferroptosis. Now that sounds technical, but honestly, the best way to think about it is cellular rusting. It's a special kind of cell death that gets triggered when fats inside the cell membrane start to oxidize or rust way too quickly. It causes this catastrophic chain reaction that just rips the cell apart from the inside out.
And it turns out that lipid armor the cancer cell was working so hard to build, that was the exact thing protecting the cell from ferroptosis. The armor was preventing that rusty chain reaction from ever starting. So when you use an FLT3 inhibitor to take that armor away, you leave the cell completely defenseless against its own self-destruct button.
So now you have this incredible piece of information. What do you do with it?
Well, you design a smarter, more devastating attack.
The strategy is this beautiful 1-2 punch. First, you hit the cancer with punch number 1, an FLT3 inhibitor that takes down its protective armor. Then, while its defenses are down, you come in with punch number 2, a totally different drug that's designed specifically to kickstart that rusty fat process of ferroptosis.
It's a brilliant strategy, right? You use one drug to create the weakness, and then a second drug comes in to exploit it. The hope here is that this kind of combination therapy will be way more effective at destroying these cancer cells than either drug could ever be on its own.
You know, this whole discovery is such a powerful reminder that the key to beating cancer often lies in just understanding its most basic survival tricks. By figuring out the exact molecular machinery a cancer cell uses to build its armor, we can find the blueprints for its own destruction. It really makes you wonder, doesn't it, what other cellular secrets are out there just waiting for us to find them?